Hello my beautiful students, so welcome back to the KCET Masterstroke series. So here we are going to discuss the biomolecules chapter. I guess whatever the previous chapter we discussed, you are going through it, and please don't forget to comment me below if you are having any issues. So today we are going to discuss about the biomolecules. So yes, let's proceed. So here we are going to cover carbohydrates, protein, enzyme, vitamins, nucleic acid and hormones. So here first we will start with the carbohydrates. So in carbohydrates we will start with classification. Here we will discuss monosaccharides, disaccharides, polysaccharides. In monosaccharide we will discuss the classification, preparation of glucose, structure of glucose, cyclic structure of glucose, structure of fructose. Next, in disaccharide we are going to discuss sucrose, maltose, lactose. In polysaccharide we are going to discuss starch, cellulose and glycogen. So yes, let's proceed with the, here you can see classification part we will proceed first. So here you can see first in carbohydrate we can find out, we can classify the carbohydrate with respect to the product of hydrolysis and with respect to the reducing nature. So in product of hydrolysis we can get monosaccharide and oligosaccharide and polysaccharides. In reducing nature we can get reducing sugars and non-reducing sugars. So yes, first we will discuss about the monosaccharides. Here also classification comes. Monosaccharides can be classified with respect to the number of carbon atom and functional group. In accordance to the number of carbon atom, we can get triose, tetrose, pentose, hexose, heptose. Each represents the number of carbon atom present over there. And in accordance to the functional group, either it can be having aldehyde or it can have the ketone. So aldose or ketose with respect to that. Let's proceed. Here from sucrose, as a preparation of glucose, if we proceed from sucrose, we can do by hydrolysis of sucrose we can get fructose and glucose. From starch what we can do, let's say here also hydrolysis we can perform, and whatever the number of N, like whatever the number of carbon it's present over here, that many unit of glucose we will get out of it. Okay, next here you can see first is molecular formula of glucose. I guess you know C6H12O6. I don't have to tell you this. Reaction with HI, this is a very important thing. Why? Because entire structural analysis of glucose, they give small question out of it in your board examination, as well as in KCET also the PYQ. If you follow, you will get the questions coming from this zone. Okay, so a reaction with HI. Reaction with HI will prove you that yes, my glucose actually contains 6 carbon. Okay, because if you treat glucose with HI, you will get N-hexane, a chain of 6 carbon. That proves that yes, all the 6 carbon are linked in a straight chain, and carbon and the glucose is containing 6 carbon system. Let's proceed. Here we are going to discuss the reaction with NH2OH, that is hydroxylamine, and HCN. So if you give NH2OH and then it will create CHNOH, and then if you give HCN, it will give you the cyanohydrin type of compound that confirms that glucose is having an aldehydic unit over here. Okay, now so this particular thing will give you the idea of presence of a carbonyl group at first. Okay, but now the carbonyl can be of different type. It can be ketone, it can be aldehyde, it can be carboxylic acid, right? So many things. But here what type of carboxylic acid cannot be there actually. Here what type of carbonyl compound is there? For further, if you just treat this with bromine water, okay, then it will end up with the gluconic acid. Okay, that gluconic acid will confirm that the carbonyl group that is present, it is basically the aldehydic unit. Okay, let's proceed. Okay, so here we are going to discuss about the reaction of acetic anhydride. So if you see, if acetic anhydride reacts, you will get this type of thing that will prove that the presence of 5-OH group. Okay, this 5-OH group should be attached to different carbon. Okay, means not in a single carbon I'm having all the more than one OH group. Rather, this 5-OH group, they are attached in different compound. Okay, so This is confirmed whenever you are treating this with acetic anhydride. Okay, now if you react this with HNO3, then oxidation takes place. Okay, oxidation takes place and you will end up with the saccharic acid and as well as the gluconic acid. It will prove that whatever the OH group you are having, all of them are primary OH, primary hydroxyl group. Okay, so I guess you understood this part. Let's proceed further. So this is the representation of D-glucose. Now limitation, it doesn't react with Skiff base because it is having aldehydic unit. It should react with the Skiff base and the reagent like NaHSO3. It doesn't react with it, but as aldehyde unit is present, it should react. That is the limitation. Okay, now pentacetate does not react with NH2OH, indicates the absence of free CHO group. Okay, now it exists in two different form, alpha and beta, that also cannot be explained. Now here you can see it was proposed that one of the OH group may add to the CHO and form cyclic hemiacetal structure, means one OH and that CHO that is at the top, they can attach to form a hemiacetal like linkage, and this particular thing explains the absence of CHO group and also existence of glucose in those alpha beta two different form. So these are the limitations. Okay, let's proceed further. Here this is the alpha beta form. You can see this carbon 5 OH can make hemiacetal like linkage with the aldehydic carbon and it can form the beta. Okay, so this particular thing actually explains the existence of those alpha and beta forms. Okay, I guess it's clear to you. Let's proceed further. This is alpha and beta, two different form, I already told you. Just it is with respect to the anomeric carbon. This carbon is called anomeric carbon, this carbon 1. With respect to this, OH is top, it is beta, OH is below, it is alpha. Okay, on being proceed further. Okay, this is the pyran ring, fine. Here you can see we are going to discuss about the monosaccharides. Okay, so here in monosaccharides we can discuss about the fructose. This is the structure of fructose, you can see. So fructose also, it can form fructofuranose by making the bond with the alcoholic unit and this carbon 5. Okay, aldehydic carbon and carbon 5, they can form acetal like of linkage and it can form fructofuranose type of ring. Okay, so this is also alpha and beta, two different form, it also exists. In the anomeric carbon, here is 2, carbon 2 is anomeric carbon. With respect to carbon 2, CH2OH is top, CH2OH is below. Okay, so I guess it's clear. Okay, one more thing that I wanted to convey to you people, that yes, our Deeksha Karnataka channel already has launched ABHYAS KCET test series. Please enroll yourself as soon as possible to check your ability, your position, in order to check the preparation in KCET level. So yes, along with that, they already have launched the KC30 batch as well. That KC30 batch is also a little bit premium, that will give you some live online session by the expert faculties, previous year questions, complete syllabus revision along with the PU1 complete syllabus, chapter wise KCET PYQ in test format, and as you can see the offer, that is 2499, it's valid only till 25th Feb. Please make sure that you are taking this opportunity and you are making your effort to the top level. So yes, don't forget, enroll yourself right now. So yes, let's proceed further. Here we are going to discuss about the disaccharide. So sucrose on hydrolysis, it gives the equimolar mixture of glucose and fructose. Okay, so as you can see, on being hydrolyzed, it will give glucose and fructose. Okay, now these two monosaccharides are held together by a glycosidic linkage. So what is glycosidic linkage? Basically C1 of alpha D glucose and C2 of beta D fructose. Okay, in between them, if one unit of water is released, one glycosidic linkage like this, it is formed. They are linked together in sucrose molecule. So whenever you are hydrolysis, means you are putting water, you are giving back the water, then definitely this glucose and fructose will be separated like that. So this is a non-reducing sugar. Okay. So now one phenomenon that I want to explain, that is the inversion. So basically sucrose is dextrorotatory, but whenever it's being hydrolyzed, it will give you dextrorotatory glucose and levorotatory fructose, right? But then why I am observing sucrose as dextrorotatory, right? So what is the reason? The levorotatory fructose is more than that of the dextrorotatory glucose. So what is happening? The levorotatory fructose is more than that of the dextrorotatory glucose. If you see the levo-rotation, it is minus 92.4 almost, and if you see the dextro-rotation, it is plus 52.5. So levo-rotation overcomes the dextro-rotation of glucose. As a result of this, the mixture after hydrolysis, you are getting levo-rotatory. Okay, so thus after hydrolysis, sucrose gives the solution that shows the levo-rotation, right? So what is the overall thing? Sucrose is dextrorotatory, but once it's getting hydrolyzed, you will get the solution that is levorotatory. Why? Because on being hydrolyzed, it is giving levorotatory fructose and dextrorotatory glucose, but levorotation is more than that of dextrorotation. It is overcoming the dextrorotation. Okay, as a result of this, the mixture of the hydrolysis becomes levorotatory. Okay, so this particular thing, that this change in the sign of rotation, that is from dextro to revo, and the product is named as invert sugar. Okay, this phenomenon is called inversion and the product is named as invert sugar. Okay, I guess it's clear to you. Let's proceed further. Here you can see we are going to discuss about the disaccharide, that is also maltose. Okay, it is composed of alpha D glucose, that is carbon 1 of glucose is linked with carbon 4 of another glucose. So carbon 1 of alpha D glucose and carbon 4 of another glucose. Okay, so it's a reducing sugar. Next you can see we are going to discuss beta galactose. Okay, we are going to discuss about lactose. We are going to discuss here about the lactose. It is composed of, please remember, maltose, lactose, this particular thing sometimes they ask to draw the structure in Haworth projection. Okay, so this is how you have to represent the structure in that case. So lactose is a combination of beta D galactose and alpha D glucose, beta D galactose and alpha D glucose. So the linkage is between carbon 1 of galactose and carbon 4 of glucose. Okay, they are attached with the glycosidic linkage. This is reducing sugar. If I proceed, then polysaccharide we can discuss about the starch, amylose, amylopectin. Okay, so like here what we can discuss about amylose and amylopectin, what is the difference between them? So amylose is basically water soluble, amylopectin is insoluble. Amylose contains almost 15 to 20% starch. Okay, amylopectin, it contains 80 to 85% starch. It is amylose, if you discuss, it is unbranched, wherever amylopectin is branched. Okay, and if you discuss about amylose, alpha D glucose units, they are in between carbon 1 and carbon 4. Okay, it is linkage alpha D glucose to alpha D glucose, they are linked by carbon 1 and carbon 4. Okay, so now amylopectin, if you look at, then it is a polymer of alpha D glucose unit. Definitely glycosidic linkage in between carbon 1 and carbon 4, but the branching takes place in carbon 1 and carbon 6. Always remember, the glycosidic linkage, it forms in between carbon 1 and carbon 4 of the alpha D glucose, but whenever it comes to the matter of branching, it takes place in carbon 1 and carbon 6. Okay, so I guess it's clear. Let's proceed. So this is the amylose, this is how it looks like, this is not branched at all. Now here comes the amylopectin. Here you can see carbon 1 and carbon 4, they are making the glycosidic linkage, and carbon 1 and carbon 6, they are making the branch. Okay, I guess it's clear to you. Let's proceed. Cellulose is definitely it's a straight chain. It's a polysaccharide of beta D glucose. Okay, definitely the linkage is between carbon 1 and carbon 4. Where do we see this? Definitely in the cell wall in plants. Okay. Okay, so let's proceed further. Here you can see we can discuss about the glycogen. These carbohydrates, they are stored in animal body as glycogen. Okay, this is also called animal starch, and it's very branched structure. It is usually present in our muscle, in our brain, majorly in liver. Okay, so whenever body needs glucose, then enzyme breaks down this glycogen and it gives us the extra glucose supply. Okay, it is also found in different type of yeast and fungi as well. Okay. Here comes the second part of the biomolecules, where we are going to discuss about the proteins and enzymes. In proteins, we will discuss about amino acid, classification, structure of proteins, denaturation of proteins. Okay, so let's start. Here comes the alpha amino acids. Okay, amino acid means we are having the NH2 group and COOH group in the same carbon. Okay, so here comes, you can see there will be a side chain. Definitely with respect to the side chain, there comes the different type of amino acid. You can see glycine, alanine, serine, threonine, different type of name of amino acids are given here along with their formula. Okay, let's proceed. Here comes few more. Okay, so let's discuss about the Zwitter ion. So what is Zwitter ion? Basically, I am having the same unit, means same one basic unit, one acidic unit in a same compound. Acidic unit is COOH and basic unit is NH2, right? So what basically they do, carboxylic group, it releases its hydrogen and the base takes up that hydrogen. Okay, so COOH becomes COO minus and NH2 becomes NH3 plus. Okay, so this charge separated version, when they exist like this, we call it Zwitter ion. Okay, this is neutral in nature, but it is containing negative and positive charge in its two end. Okay, and it is definitely amphoteric. Okay, this is called Zwitter ion. Okay, so now here comes the classification. You can see amines can be natural and synthesis. Natural, you can see acidic, basic or neutral, and in synthesis basis it can be essential and non-essential. Okay, so here comes the example of some essential amino acid. You can see valine, leucine, isoleucine, arginine, lysine, threonine, methionine, phenylalanine, tryptophan and histidine. Okay, so now here you can see how the peptide linkages are formed. Okay, so amino acid, they are actually connected to each other with the peptide linkage. So one COOH means one carboxylic acid end and another NH2 end of another amino acid. If one unit of water is released, then they form the peptide linkage, CONH. This is called the peptide linkage. So here you can see one glycine and one alanine. So glycine glycine carboxylic acid unit and alanine NH2 unit, they are one unit of water is released and they are forming the peptide linkage. Okay, so when the number of such amino acid is more than 10, the products are called polypeptides. Okay, so more than 10 amino acids are actually attached with each other by the peptide linkage, then they are called polypeptide. Okay, so a polypeptide with more than 100 amino acids residue having the molar mass almost 10,000 is called a protein. Means it's a very long chain, okay, of polypeptide. I guess it's clear to you. Now if we discuss about the structure of protein, it can be of molecular shape. In molecular shape you can have a fibrous and globular, and in structure you can have primary, secondary and tertiary, quaternary. Okay, so fibrous and globular, it's a very important zone for the board as well as for the KCET. Okay, so like what is fibrous protein and what is globular protein? So fibrous proteins are basically polypeptide chain, they run parallel to each other, okay, and they are held together by hydrogen bond. But whenever it comes to the matter of globular protein, it's basically coil like system, spherical shaped. Okay, fibrous protein, they are insoluble in water. Globular protein, they are soluble. One fibrous protein that is there present in our hair, that is keratin, or you can see any kind of silk, wool, like that. Globular protein, if you see insulin, albumin, this type of things. Okay, I guess it's clear. Let's proceed. If you just look at like primary structure, then primary structure is basically sequence of amino acid, and whenever it comes to the matter of secondary structure, two type of thing we see, alpha helix and beta plated sheet. Okay, so alpha helix is basically due to regular folding of the backbone. The backbone will be just folding, just if you keep folding the papers, okay, one by one, one after one, like that. Okay, so this will give you definitely the folding is held by the hydrogen bonding only, hydrogen bonding in between CO and NH groups. Okay, so this is basically alpha helix. And beta plated, if you see, this is basically the polypeptide chain, that is they are held side by side. Okay, and definitely here also intermolecular hydrogen bonding takes place. Okay, this is how the structure looks like. You can see this is my alpha helix and this is my beta plated sheet. Okay, I guess it's clear to you. You can see clearly the hydrogen bonding, how it's created. Okay, let's proceed. Okay, so here we are going to discuss the tertiary structure and quaternary structure. So tertiary structure, it represents overall folding of the polypeptide chain, further folding of the secondary structure. Okay, it can give you the fibrous or globular. Okay, definitely different type of interaction can make it stabilize, such as hydrogen bonding, van der Waals interaction, electrostatic interaction, they can stabilize the structure. Okay, what is quaternary structure? It's a definitely a 3D arrangement, where two or more polypeptide chain, when it actually special arrangement, or two or more polypeptide chains referred as subunit that repeats each other. Okay, like the example is hemoglobin. Okay, that is quaternary structure. A little bit more complex, more complicated structure. Okay, I guess it's clear to you. Let's proceed further. Here you can see the overall thing. This is a primary, is just the repeating unit. This is a secondary, alpha helix and beta plated sheet. This is a tertiary, and this is a quaternary. Okay, more subunits are repeated here. Okay, it's even more complicated thing. I guess it's clear to you. Let's proceed further. So here comes the denaturation of protein. So here you can see what is denaturation. Different type of physical change, like if you change the temperature. Okay, if you do any kind of pH change, means chemical change, then what happens, the hydrogen bonding inside the protein, they gets disturbed. Okay, and the globules, they unfold. Okay, the helix, they gets uncoiled. Okay, as a result of this, protein loses its biological activity. Okay, so this phenomenon is called denaturation of protein. In 1 degree protein, there is, in primary protein, there is no as such denaturation, but in other cases, when the structure is complicated, in those cases the hydrogen bond will be disturbed because of the change in different physical and chemical conditions, right? Like coagulation of egg white, right? Curdling of milk, this type of thing is called denaturation of protein. Okay, let's proceed. Enzymes are called biocatalysts. Okay, they are generally globular protein. They are very much specific towards its substrate. Means if you just see, if here this particular enzyme, if you wanna put, it will not work here. But if here in this particular substrate, this and this will adjust nicely. But here if you want to put this circle one, it will not adjust. So only specific enzyme like this and this will only work in this substrate. So all the enzymes are very much substrate specific. Okay, it definitely it works similar to that of a catalyst that reduces the activation energy. Okay, so one question you can see, proteins are found to have two different types of structure, secondary structure, that is alpha helix and beta plated sheet structure. Alpha helix structure of protein is stabilized by, definitely it's going to be hydrogen bonding. Okay, let's proceed. This is vitamins. What is vitamins basically? It's an organic compound that is required in your overall diet in a very small amount, but it performs a very specific biological activity to maintain your normal optimum growth and health condition of an organism. So it's very important. Okay, so vitamins, you can see it can be fat soluble and water soluble. Fat soluble is basically that is soluble in oil, insoluble in water, that is ADEK. So they are usually stored in our liver and in adipose tissue. Okay, and another one is water soluble, that is vitamin B and vitamin C. They should be taken regularly. Okay, so here we will discuss one by one the vitamins, the source, and in absence of them what kind of disease or what kind of thing you can observe out of it. So vitamin A, you can get it out of fish liver oil, butter, carrots, milk, and in presence, in absence of or in deficiency of vitamin A, the disease that is called xerophthalmia. Okay, that is basically the hardening of the cornea. Okay, night blindness you can expect. And vitamin B that is also called as thiamine, we can get it out of yeast, milk, different type of cereals, green veggies. Okay, so in absence of vitamin B, you can expect berry, that is a lot of appetite, and it is definitely related to the growth of an organism. Okay, next vitamin B2, that is also called as riboflavin. You can get it in milk, in egg white, in liver, kidney. Okay, and in absence of this, you can expect chelosis. Okay, you can expect chelosis, that is basically fissuring at the corners of the mouth. Okay, fissuring means just at the corner of the mouth there should be some ulcer type of things. Okay, some digestive disorder can be observed, some burning sensation can be observed like that. Okay, next here you can see vitamin B6, that is also called as pyridoxine. It is we can get it out of yeast, milk, egg yolk. Okay, cereals. Okay, so in absence of this, you can get some you can expect convulsion. Okay, vitamin B12 we can get out of meat, fish, curd. Okay, in absence of this, you can expect anemia, that is RBC deficiency. Okay, in hemoglobin. And next is vitamin C, that is none other than ascorbic acid. You can get it from different type of citrus food, amla, green leafy vegetables, and in absence of this, you can get scurvy. Okay, that is in general we will call it bleeding gums. Okay, next vitamin D. Vitamin D, definitely one of the major sources, sunlight, right? Exposure to sunlight, fish, egg yolk, we can get the vitamin D. In absence of vitamin D, definitely we can get rickets, that is different type of bone deformities in children. Okay, and we can also observe osteomalacia. Okay, so osteomalacia is something like softening of bones. Okay, softening of bone joints mainly in the adults. Okay, so this is vitamins. Now fat soluble, you can say vitamin E. Vitamin E we can get in some vegetable oil, sunflower oil. Okay, so this is basically in absence of it, we can face some muscular weakness, some fragility of the RBC will be increased. Okay, next vitamin D, vitamin K, if you see, that we can get out of green leafy vegetables, and it is very much helpful for the blood clotting. Okay, so okay, this much. Let's proceed with the nucleic acid. So what is nucleic acid? Nucleic acid is a composition of pentose sugar, phosphoric acid and nitrogen containing heterocyclic compound majorly called as base. Okay, so nucleic acid is made up of pentose sugar, one phosphoric acid and one nitrogen containing base. So pentose sugars are RNA and DNA, ribonucleic acid and deoxyribonucleic acid. You can see the structure over here. And nitrogen containing base, that is also different in RNA and DNA. Okay, we will see different bases over there as well. Okay, so now here you can see nucleoside is basically, what is nucleoside? Nucleoside is basically one nitrogen base plus the sugar. Okay, so the unit formed, how it is formed, like the one position of the sugar is attached with the base. Okay, one position of the sugar, one carbon of the sugar is attached to the base. This is called nucleoside. When these nucleosides are linked with the phosphoric unit at the fifth position of the sugar moiety, so this nucleoside further they are getting attached with the phosphoric acid, where at the fifth position of the sugar, then we call it nucleotide. So nucleoside is the monomer. Then nucleoside, when they are connected by phosphoric acid, then we will call it nucleotide. Okay, so these nucleotides, they are joined further with the phosphodiester linkage. Okay, that is between carbon 5 and carbon 3 of the pentose sugar. Okay, so here you can see in the fifth position, you can see in the carbon 1 the base will be attached. This is the nucleoside. Okay, and here you can see in the fifth position, in the 1 position as usual I am having base, this is my nucleoside. In the fifth position I will be having the phosphoric acid unit. Okay, let's proceed. So like this is one nucleoside, this is one nucleoside, this is one nucleotide, this is one nucleotide, right? Further they are attached, this phosphoric acid unit they are attached with the phosphodiester linkage, right? They are further attached with the phosphodiester linkage. Okay, so I guess it's clear. Let's proceed further. Nucleic acid also can be a primary structure and secondary structure, like sequence of nucleotides in a nucleic acid. That is basically the primary structure. And secondary structure, if you see, that is the two nucleic acid wound about each other and held together by the hydrogen bond. Means two nucleic acid unit, here one, they are held together by the hydrogen bond. Okay, so two strands, they must be complementary to each other, means one, if it goes upside, one must go downside. Okay, so now here you can see adenine forms hydrogen bond with the thiamine, whereas cytosine forms the hydrogen bond with guanine. Always remember ATGC. Okay, adenine forms the bond with the thiamine and cytosine form the hydrogen bond with guanine. Okay, so this is the DNA structure. This is how the double helix it is basically formed. Okay, so here in the nucleic acid, what is the main role? It stores the genetic information and it is responsible for the protein synthesis as well. Okay, so yes, this is a function. Let's proceed with the hormones. Okay, so hormones are called intracellular messenger. Okay, and it is secreted by some endocrine gland inside our body itself, and it is poured directly to the bloodstream. Okay, so now different type of hormones we are going to discuss with example. Like you can see polypeptide type of hormone, means it's protein, right? So insulin, endomorphins, glucagon. Okay, now here you can see amino acid and its derivatives. It is basically epinephrine, norepinephrine, thyroxine. Okay, next steroidal hormone, that is estrogen and androgens. Different type of hormones. Okay, next you can see here we are having some set of hormones, and here we are having some functions of it. Okay, so here you can see first we are going to discuss about the glucagon. So glucagon is responsible to increase the glucose level in your blood. Next, insulin, it is just opposite to glucagon, it maintains the sugar level in your blood. Next, epinephrine and norepinephrine, this is responsible to response to external stimulation of your body. Next, thyroxine. So thyroxine, it is usually produced in your thyroid gland. So iodinated derivative of amino acid tyrosine. What is thyroxine? This is basically a iodinated derivative of amino acid tyrosine. Okay, so whenever I am having low concentration, I will be having hypothyroidism. Okay, if I am having low concentration of thyroxine, then I will be having hypothyroidism. Okay, so that is basically, that leads to the lethargies, obesity. Okay, and if you are having high concentration, that is hyperthyroidism, that is enlargement of the thyroid gland. Thyroid glands are situated here, just two sides of your larynx, right? So if it gets enlarged, you can further observe goiter. Okay, so this type of thing. Let's proceed. We are going to discuss about the steroidal hormones. They are majorly produced by adrenal cortex and gonads. Okay, we are going to discuss glucocorticoids first. So it is definitely released by adrenal cortex. It actually control the carbohydrate metabolism in your body. It modulate the inflammatory reactions. Okay, so any kind of inflammation or something if you see in your body suddenly, it is maintained by the glucocorticoids, and it is also involved for your stress. Okay, next, mineralocorticoid. This control the level of excretion of water. Okay, and salt also by the kidney. Next, you can see Addison's disease we are going to discuss. It is basically the malfunctioning of the adrenaline cortex. So once adrenaline cortex will be malfunctioned, then glucocorticoids and mineralocorticoids, they will come in a abnormal way as well. That is called Addison's disease. Okay, so as a result of this, you can see hypoglycemia, okay, weakness and increased susceptibility to stress. Okay, I guess it's clear. Let's proceed. Okay, here we are going to discuss about the testosterone. Okay, so testosterone, definitely it's released by the gonads. It's a major sex hormone produced by the male. Okay, it is responsible for the development of the secondary male character, like very deep voice, hair growth, okay, and different type of physical general physical constitution. This type of thing is done by testosterone. Next, estradiol, it's a female sex hormone. It is also responsible for the development of the secondary female character. Okay, we can discuss about the menstrual cycle here. Okay, and progesterone, it is responsible for the preparation of the uterus for the implantation in the future. Okay, implantation of the fertilized egg. Okay, that is done by progesterone. Okay, let's proceed. Yeah, that's it. I guess we covered the entire thing, whatever the probable question zone that also I told over here, and definitely we will come up soon with the previous year questions. We will discuss thoroughly, and by that time, please prepare this chapter, make a proper note out of it, and if you are having any doubt related to the chapter, please don't forget to comment me below, and finally don't forget to like, share and subscribe to get such informative videos. Thank you so much for watching.